Yeah. As if you are completely just bored and have no idea what to do. All right. So we have a few more questions that we need to deal with. We've already dealt with what's unique about eukaryotic cells, and the main difference is the presence of the nucleus. And so we need to talk about what is it about that nucleus that's so important. So important. The biggest one is that now you can separate the genetic material from the cytosol. And that's really important because now the cell can start to make enzymes that chew up nucleic acids, right? Because if we're talking about nucleic acids can be used as energy, like our other biomolecules, you need enzymes that can chew up those nucleic acids. Well, if your genetic material is sitting in the same place those enzymes are, you have a problem. Would you agree? And so having a nucleus actually allows the cell to make a lot of those enzymes so it can harness energy from nucleic acids. Now, the membrane is reinforced. It is a very, very strong membrane. And so that's one benefit of it. It allows the nucleic acids to be separated from the cytosol, from the cytoplasm. But another benefit of that is it makes it to where it's really hard to get access to the genetic material. It's really hard to get access to the genetic material, which actually lowers mutation rates in eukaryotic cells. So mutation rates in eukaryotic cells are lower than mutation rates in prokaryotic cells. Okay? Again, mutation rates in eukaryotic cells are lower than mutation rates in prokaryotic cells. But you still need to be able to get into and out of the nucleus. So within this membrane, there are nuclear pore complexes made of proteins that allow materials to leave the nucleus in the case of RNA, and then also allow materials to enter the nucleus, RNAs and proteins. Okay, so that's part of what's unique, but now what's a problem? If you need something in the nucleus, remember we left off last time telling you that you shouldn't think of the inside of a cell as just this massive underwater just wasteland, but instead as a very highly organized structure like an underwater city. And so if you're going to need to get something to the nucleus, you're not just going to passively wait for it to get there. Instead, you need to ensure that it gets there quickly. And how do cells do that? Here's how the cells do that. So material that needs to function in the nucleus has what's called the nuclear localization signal. So it has a piece at the beginning of that material that transport proteins that go to the nucleus, recognize, grab onto it, and take it to the nucleus. And you're going to start to see this as a theme, that materials, not only do they have what's in them to make them work the way that they're supposed to, but there's also some kind of a signal in materials as well, like a label telling it where it needs to go. Okay? So there are basically two major parts of any biomolecule working in the cell. There's the piece that allows it to carry out its function, and then there's the piece that provides the label, telling the cell where it needs to go and helping it to be transported there. Okay? This is the first instance we see of this with materials that need to go to the nucleus. Within the nucleus, you find what's called the nucleolus, and this is a really dense section of the nucleus where you have an enormous amount of ribosomal RNA being produced. And we'll talk a lot more about that in a couple of weeks. But this is a major component of ribosomes. Okay, another one of our organelles. So if you were asked this question, what's unique about the eukaryotic nucleus? You can answer it functionally in that it separates the genetic material from the cytoplasm, or you can answer it structurally, right? That it's a physical barrier surrounding the nuclear material. Either way you want to answer it is fine, as long as you can provide either a good structural or a good functional explanation of what is the nucleus. So here's an image of that nuclear envelope, which is part of the endomembrane system. And then here's a zoomed in version of these nuclear pore complexes that allow materials to move through the nuclear envelope, either entering or exiting the nucleus. And as next week, as we talk about cell membranes, you'll start to realize why that is a challenging task, moving through membranes. Here's some of the research that was done on demonstrating the existence of the nuclear localization signal. And so basically what they found is when they mutated a protein or a gene to where it changed the protein at a very specific place, the protein functioned, but it didn't get to the nucleus. Okay? So a mutation in a portion of the gene that changed a very specific point of the primary sequence of this protein, the amino acid at position 128, actually kept the protein still functioned, but it couldn't get to the nucleus. And this showed that there was something within the protein itself that was a label enabling this cell to transport it to the nucleus. All right? Okay. All right. So our next question, how do cells transport materials around the cell? And you may have put as one of your functions of the endomembrane system, transporting materials around the cell, right? Any of you put that as one of the functions of the endomembrane system? Transport. So that's part of the answer to this question. How do cells transport materials? Using the endomembrane system. But it can't act alone. What else is involved in that process? The cytoskeleton. Okay, and you may have put that as one of your functions of the cytoskeleton, transporting materials around the cell. Anyways, let's deal with this question. So yeah, cells use the endomembrane system to transport proteins especially, but other materials around the cell. So the endomembrane system can also be used to bring materials into the cell. You call that phagocytosis. The cell membrane itself, the plasma membrane, is part of the endomembrane system. And so this system can be used to bring materials into the cell, and it can be used to release materials from the cell. You call that exocytosis. And let's take this name apart. So we have the endomembrane system. What does endo mean? Inside. What does membrane mean? Yeah, like a covering, right? It means a membrane, right? So we're talking about a membrane, a system of membranes within the cell, and that's exactly what the endomembrane system is. It's a series of membranes. Some of them are directly attached together, but many of the times they are not physically attached together. But you've got these series of membranes used to transport proteins and other materials around the cell. So here are your components of the endomembrane system: the nuclear envelope, the endoplasmic reticulum, Golgi bodies, lysosomes, vesicles, and the plasma membrane. Okay? Again, these series of membranes, either physically or indirectly attached together, functioning to move materials around the cell. So the endoplasmic reticulum comes in two varieties, two flavors, if you will. The smooth endoplasmic reticulum and the rough endoplasmic reticulum. And this, just to provide some context, proteins are actually modified inside of the rough endoplasmic reticulum. So another function of the endomembrane system is protein processing. I don't know if you put that as one of your functions of the endomembrane system, but protein processing is a really important function carried out by the endomembrane system, specifically within the endoplasmic reticulum. And again, we'll talk more about this in a couple of weeks when we talk about making proteins and what happens. What does that actually mean? Yeah. Sorry, I was seeing what comes next. Lipids are made in the smooth endoplasmic reticulum and in cells that are exposed to large numbers of toxins. There are machines within the smooth endoplasmic reticulum that destroy toxins as well. What types of cells in our bodies are exposed to high levels of toxins? Yeah. The liver cells. And so liver cells, the smooth endoplasmic reticulum of liver cells functions almost entirely in detoxification, just detoxifying materials. All right, and then proteins are further modified and packaged for delivery in the Golgi apparatus or the Golgi body or the Golgi complex, whatever you want to call it, all names for the same structure. And finally, vesicles carry materials between structures in the endomembrane system. But again, I don't want you to think about this as like a boat or a submarine aimlessly traveling through this aquatic environment. It's very highly organized. Vesicles only move in a specific direction and are labeled to ensure that they are moved in a specific direction. Okay, it's all very, very highly organized. Very highly organized. All right, any questions about the endomembrane system or moving materials around a cell?
Can you stay in this like 3 seconds? Sorry.
Maybe even more than 3 seconds. Yeah, Joe.
So with like the packaging of the Golgi bodies, they say that like, I mean, we kind of seen the whole process. Like one cell makes protein, gets packaged, released outside of the cell, and then when it bumps into another cell, those like tags you were talking about that get things in the nuclear envelope, are those the same tags that like would have been placed like by the Golgi body?
Yes. Yep.
Okay.
Yeah, a lot of times the final processing of a protein involves adding that label. Yeah. Adding that label that ensures the protein is transported to the correct place. And it's not just proteins needing to be transported to the nucleus, right? Some proteins function inside of the mitochondria. Some proteins function inside of the Golgi apparatus. So they get finally processed in the Golgi apparatus, and then they stay there because they function within the Golgi apparatus. Yeah, so part of that final processing is oftentimes adding the label. So sometimes it's not actually originally part of the primary sequence, but instead is something that's added on a tag. Or it can be part of the original primary sequence of the protein as well.
So a mailroom really is like a good analogy for that then.
Oh, absolutely.
Okay.
Yeah, absolutely.
Okay.
Yeah. Now the big difference is in a mailroom, you're really just, transferring materials, right? They come in, you move them somewhere else. Whereas in the Golgi apparatus, there's like final modification too. So it'd be like, if you order something that's not fully assembled and it goes to your mailroom and some really kind worker there assembles it for you and then takes it to you. So it's like the mailroom, it's like the way my children experience things being shipped to them, right? It comes to me, I put it together for them, and then I give it to them. Yeah,
What defines something in the endomembrane? Because aren't there things that have a membrane but aren't part of the endomembrane?
Oh, sure. Yeah. The mitochondrion has a membrane and is not part of the endomembrane system. And so it's typically a function. It's a functional issue, whether or not you would consider something to be part of the endomembrane system, and not a structural issue of having a membrane, right? If it's functioning in this process of processing and transporting materials, then it's part of the endomembrane system. If it doesn't function in that, then it's not. But it still needs to get materials from the endomembrane system. Yeah, Caleb.
Is there a video you would recommend to help us visualize this?
I can find one. Why, do you know of one?
Well,
You asked that with like a knowing smile.
well, because my dad teaches science, and so he has videos, but I vaguely remember him showing me something when I was little, and I don't know if
Here's the thing about videos is they're nice, but oftentimes they're not perfect, and so then I get angry. And but I'll try to find one. I don't mind doing that. So very quickly, I just want to show you a process of the endomembrane system, and then we'll take ourselves a lecture break. All right, so here you have the nucleus, the nuclear envelope, which is actually continuous with the endoplasmic reticulum. The membrane is continuous. So these are both part of the endomembrane system. Here you can see vesicles connecting the endoplasmic reticulum to the Golgi body. They are not physically connected. They are indirectly connected through vesicles, and then more vesicles actually taking materials from the Golgi body outside of the cell, taking them to other places. And what's actually neat is these what's the term? I can't even remember the term for these individual sacs. No, the Golgi apparatus is made up of a series of sacs, and for some reason I can't remember the specific term of that. Anyways, it'll come to me. But even the Golgi apparatus is not one continuous membrane. It's a series of sacs that material transfers in between them through vesicles. All right, so we're going to take ourselves a lecture break. And what I want you to do is now with your discussion group, I want you to do something together, and that's to take your endomembrane system and provide the missing parts. Okay? So this diagram is helpful, but it's incomplete. There's something missing here. And I told you a little bit ago that these vesicles do not move in random ways. They move in a specific direction and can actually only move in a specific direction and on something. And so there's something missing from this diagram. And so what I'd like for you to do is take a couple of minutes working with the people in your discussion group, and then we will talk about it collectively to explain what's missing. All right? Take a couple of minutes starting now.
Hamburger and french fries.
The peroxy stuff.
You mentioned that so we say the cytoskeleton.
Doesn't say word.
That's in the nucleus. I think the endomembrane system is more like the.
Well, it technically happens because the ribosomes it's all in there.
Mitochondria. Does it say cytoplasm?
Yeah.
So it's cytoplasm.
You got
Do they need energy from it, or is it just like set
I mean, they have the vesicles.
technically, yeah.
So they need
Was the plasma membrane there? Obviously. No, there's no reason. Well, that's part of like the cytoskeleton. I think that's kind of what
All right, take another 10 seconds or so, wrap up your thoughts. All right, group one, what's missing? Yeah, you are group one.
the cytoskeleton.
Okay, cytoskeletons, any specific parts of it that would be really, really helpful in the explanation of this diagram? Or just generally cytoskeleton?
Well, I don't know the specific names, but
Okay.
there's like little like fibers, like
Uh-huh.
ways that the vesicles travel along.
Yep, absolutely.
Is it microfibers? Or microfilaments?
Nope, not
I
microfilaments.
don't know the name.
Yeah. What do we got? Yeah, Daniel. Microtubules. Yeah, and so you have three categories or three elements, if you will, of the cytoskeleton. You have microtubules, you have intermediate filaments, and you have microfilaments. And the microtubules are the major piece that's missing from that diagram on the endomembrane system, because it gives you kind of this idea that their vesicles are moving kind of randomly and are moving just inefficiently or by diffusion. But that's not the case at all. They are dragged along the microtubules to their particular destination. Yeah, Charles?
So two questions. Number one, microtubules are hollow?
Yeah, sort of.
Travel inside the microtubule?
No. You know, there are motor proteins that attach to the outside of the microtubule and attach to the vesicle. So the motor proteins walk along the microtubules while holding on to the vesicles. Yeah. And the microtubules are sort of hollow, but I mean, functionally, the proteins take up more space than what it seems. But yeah, I mean, they're sort of hollow. But yeah, motor proteins kind of walk along the microtubules while holding on to the vesicles. I mean, they don't kind of do that. That's exactly what they do. They walk along the microtubules while holding the vesicle. Yeah. And so you have motor proteins that go in specific directions.
So how does that work? Specifically?
We'll talk about that. Okay. Yeah, we'll get there. Yeah. Not right now, though. Right now, we're just laying a foundation. So we'll get into those details. Yeah, Lacey? Okay. All right, so the last four questions that we need to cover in our discussion of chapter 4, and I know chapters 2, 3, and 4, they are frustrating because it's like, I mean, they're just introducing topics and you don't really get to talk about them very much. And then if it's not really something you've covered before, it just I mean, it's like you're jumping around on a map with no indication of where you are, right? It's like here's your progression, right? Like your pit stops on a road trip, but you have no idea what's actually going on. Yeah, it's frustrating, but we'll start to fill in those gaps. All right. So the fourth to last question: how do cells generate energy? And so you have this process called cellular respiration, which we'll spend an entire week. Maybe even more, depending on how our conversation of cellular respiration goes, talking about cellular respiration and what's actually going on. But this happens partly inside of the mitochondrion. The one part of cellular respiration takes place in the cytoplasm, but the bulk of it happens inside of the mitochondria. And so each mitochondrion has a double membrane. And so what it allows you to do is to set up really steep concentration gradients. And we haven't really talked a ton about diffusion yet. We will next week. But when you set up a steep concentration gradient, materials want to move from where they're highly concentrated to where they're less concentrated. And so if you set up a really steep concentration gradient, which we do in between these two membranes, packing an enormous amount of hydrogen ions, the hydrogen ions want to go flying back to where there's very little of them. And when they do, you can harness that energy and use it to actually do stuff. It's nice. So the inner membrane encloses what's called the matrix. And then you have these folds within the inner mitochondrial membrane that almost produce like disc-like structures within the mitochondrion that we call cristae. And so inside of the matrix, you have mitochondrial DNA, you have ribosomes, you have other components that function inside of the mitochondrion. And the presence of DNA is interesting. So you have DNA within the mitochondrion, and what would that suggest? Yeah.
It can reproduce separately.
Yeah, that it can reproduce separately of the cell, and it can. And what else might it suggest? Yeah.
That it used to be its own entity.
Yeah, that it used to be its own entity with its own genetic code, and that's a fun one. And we'll talk through this. Again. I know we talked through this in organismic, but we'll talk through this again. So ATP is generated within the matrix in these cristae and these folds, and and generated in the folds of the cristae and inside of the matrix itself. Yeah, so the presence of DNA within the mitochondria suggests a separate origin to the mitochondria. And then you have another set of structures called microbodies. An example of this are peroxisomes or glyoxysomes or glycosomes. And these also function in either breaking down organic molecules or they function in synthesizing organic molecules for energy storage. So the mitochondrion is not the only organelle involved in energy dynamics. There are other organelles involved as well in either breaking down organic molecules to allow access to energy or in synthesizing organic molecules for storing energy. All right, make sense? Any questions? I know, again, chapter 4 is frustrating, just like 2 and 3. It's just like you're hopping all around and not really talking about anything. Yeah, Joey.
So just like we have the endomembrane system, we have like a name for it. Do we have a name for like the?
Oh, that whole complex of
all
energy.
the other ones that work with the mitochondria. Do we have
Yeah,
a name for it?
I don't, not that I know of, but it'd be nice if we
Can you
did.
give it a name?
You can
We
call it
have the
whatever you
Fred
want.
system.
The Fred system. You can call it whatever you want. And so here are the two membranes of the mitochondria. Here's the inner membrane and the outer membrane. You've got this inner membrane space. Inside of here is the matrix. And these are the folds, the cristae, that provide a great deal of surface area. Yeah.
Are the microbodies outside of the mitochondria?
Yes. Yep, they're separate organelles. Yeah, that's a good question. And so here's an electron micrograph of the mitochondria. And again, you can see these folds in the inner mitochondrial membrane. And so what it does is it makes an enormous amount of surface area. So you probably imagine that that surface area is being put to use, right? If it exists, that surface area is probably being put to use. Otherwise, why would you have those folds within the mitochondria?
Would all those spots be like the protein?
Yeah, hot spots where you've got different protein, probably protein activity, and that's why. Remember, with an electron microscopy, you are bombarding objects with electrons, and so if you have a high level of activity, you're gonna get some really intriguing things happening, and it's gonna show up as dark spots. All right, what are the components of the cytoskeleton? What are the components of the cytoskeleton? So part of your quiz question asks you for two functions of the cytoskeleton. It's helpful to then start to put in some of these structures. So I mentioned this already. The eukaryotic cells have cytoskeletons with three parts: microtubules, intermediate filaments, and microfilaments. So these microtubules, these are made from the protein that's called tubulin, which is it's a dimer, which means it's a protein with quaternary structure. Okay, dimer, di means what? Two, so this has two polypeptides that combine together to form a single functional protein. That protein is tubulin. And then tubulin polymerizes together to form these microtubules. And so arranged in 13 polymers, you have 13 long chains of tubulin that all are attached together to form these microtubules. So some of their functions, they anchor organelles in place. So what keeps the mitochondria where they're supposed to be and what keeps the endoplasmic reticulum where it's supposed to be. And these also provide the roads for these vesicles to move along. So again, these are the main thing that was missing from that diagram of the endomembrane system. So if you were to go and make that diagram better, all you would do is you would go in and you would supply the roads, right? These microtubule roadways that things can move across. They also function to separate the genetic material when the cell is dividing. We'll talk a lot about microtubules when we get into cell division because they play an incredibly important role there. Yeah, Daniel.
So the straight lines on the picture, the electron microscopy,
Uh-huh.
the mitochondria, right above it, is that microtubules
Right here?
above it?
Yeah, all of these. Yeah, it's a good question. My guess is that it's a combination of microtubules and also just picking up on different concentration differences of materials. Because there are constantly materials that are being pumped out of and into the mitochondria. And so I'm guessing that it's a combination of the microtubules and just different materials that are concentrated in bands, as they're being pumped out of the mitochondria. That's a good question. All right, next one, intermediate filaments. So these are made up of a whole suite of different intermediate filament proteins. It's not a single protein like microtubules are. And so for your benefit, we'll just leave it as a category of protein types, intermediate filament proteins. These oftentimes combine with other cytoskeletal elements. So you'll see intermediate filaments working with microtubules, working with microfilaments, the other type, to collectively accomplish some task. So these provide structural support, and we'll see this to be especially true with cell junctions. And so the last question on your quiz asked for you to provide a type of cell junction and the protein involved. And so one type of our cell junction uses a great deal of intermediate filaments to help keep cells attached together. And then the last type, microfilaments. These, like microtubules, are made from a single protein type. They're made of actin instead of tubulin. Made of actin. And these function in a number of ways. They contract muscle cells. They're involved in moving a cell using what are called pseudopodia, and that's called cytoplasmic streaming, and then also functioning to divide the cytoplasm when cells divide. Yeah, Lacey.
Actin and myosin?
Myosin's a motor protein. So actin is the cytoskeletal element. Myosin is a motor protein. Like in the microtubules, I mentioned motor proteins walking along them holding vesicles. Myosin is another motor protein, but it doesn't walk along microfilaments holding vesicles. It walks along microfilaments holding onto another microfilament so that you get, them being pulled in opposite directions and you can generate contraction. But yeah, myosin's a motor protein. Yeah. Does that make sense?
Yeah, it's usually they're classified
Together, yeah. It talks about actin and myosin in muscle cells. So actin is the cytoskeletal element, myosin is the motor protein. Yeah. So here's an image of these of a single polymer of tubulin. And so you can see tubulin as this dimer, again, a protein with quaternary structure. Two separate polypeptides functioning together to accomplish a single task. And that task is to be assembled together into a really long polymer to form a chain. Yeah?
Could that be more than a dimer since there's so many connected together?
Except for an individual dimer can exist as a unit and can function. So these dimers function separately than to just be polymerized together. But you can't polymerize just the alpha subunit. And you can't polymerize just the beta subunit. You have to polymerize them as a unit, as that dimer. Yeah. So here is an image of intermediate filaments. But again, these are extremely varied. You have a whole suite of different intermediate filament proteins. So we just say intermediate filament proteins and don't give you a specific type, tubulin or actin. And then here are microfilaments showing you actin subunits. Okay, so we still are dealing with this question, what are the components of the cytoskeleton? And we'll actually conclude our time together dealing with this question, not right now, but dealing with this question. Sorry, that was tricky. You're like, oh, sweet, it's Friday, he's letting us out 5 minutes early. It is Friday, that is true. We are not ending 5 minutes early. So motor proteins, here's your part. Motor proteins, myosin being one, moves along microfilaments, or kinesin and dynein moving along microtubules. And so that's another common theme of cytoskeletal elements, to have some kind of a structural role, but then to also have a role with motor proteins, providing a roadway for the proteins to move along. So one end of the motor protein literally walks along the cytoskeletal element, while the other end is physically attached to either a vesicle, in the case of kinesin, which is the motor protein that walks vesicles along microtubules, or it's attached to another cytoskeletal element. Dynein is an example of that, where dynein is a motor protein that walks along microtubules while attached to another microtubule. And then myosin does the same thing, walks along actin, the microfilament, while attached to more actin. And so it's a way of actually getting these massive cytoskeletal rods to move in opposite directions. So you could either generate contraction or you can generate bending and waving, which is cool because sometimes you need to generate that motion. And so here's an image of kinesin, this motor protein. Here's the stationary portion that is physically attached to the vesicle, and then here is the dynamic portion that rotates and walks along the microtubule. And so as it does, it uses energy to just keep rotating this protein around and walking along the microtubules. And you're like, wow, that seems like it would be so slow, until you see this actually carried out and moving a vesicle across an entire cell in a fraction of a second. That you're getting like tens of thousands of movements in this motor protein every second. It's just, it's boogie. It's really cool. And it's cool to actually see this model. This is one where I absolutely should get a video, because it'd be a video that wouldn't require any dialogue. And so the dialogue is usually what's incorrect and what's really frustrating. So still the same question, what are the components of the cytoskeleton? Told you we're going to be dealing with this question for a little while. We'll finish up here. So flagella and cilia are made out of microtubules that are arranged in a slightly different way. So they're arranged with 2 microtubules in the middle and then 9 triplets on the outside, or 9 pairs on the outside. It's pairs in sum. The microtubule organization center, 9 triplets at the base of the flagella and cilia. And so dynein, yeah.
That's only for eukaryotic cells, right?
Yeah, the bacterial flagella are built differently. Yeah, the machinery and the structural component is different. So dynein are motor proteins like kinesin and like myosin. These, one side walks along the microtubules, the other side is physically attached to the microtubule, and it gets them to, again, move in opposite directions. And so it can create a bend. And so you can get the entire object, the flagellum or the cilia, actually moving. So these arise from structures at the base of these. So the flagella and the cilia grow out from centrioles at their base. And these function in cell locomotion, cell movement. So another function of the cytoskeleton that you could have put on your quiz is cell movement. So you could have talked about transporting materials, cell movement, stability of the cell, protection of the cell, even cell division, all of those functions carried out by the cytoskeleton. All right? So we are out of time. You are free to go. Enjoy your weekend. You're free to go even if you're not going to enjoy your weekend. But I hope that you do. Thank you, Sydney.